Bismillahirrahmanirrahim. Welcome back, guys. Welcome to another video sessions from Chemistry Biology. In this video lecture, we will talk about the replication schemes of the retroviruses or the life cycle of the retroviruses. Now, what is retroviruses? Retroviruses are actually the HIV viruses, fine? Now, HIV viruses are those viruses which can cause the AIDS disease. So now let me write the life cycle of the. So in this video, we will talk about the life cycle of the AIDS, life cycle of the HIV. Life cycle of HIV, or we can also we can also say that replication scheme of retrovirus. Replication of retrovirus. HIV are those viruses which can cause what? Which can cause AIDS. So now let me write. It can cause what? AIDS. Fine. So it causes AIDS. How it can cause AIDS or how it can start their life cycle? So HIV contain two messenger RNA. HIV contains three types of the proteins: integrase, protease, and reverse transcriptase. Fine. So if I draw the structure of the HIV and the capsid area and the envelope area and the spikes is, so now let me draw the structure. So how it will be look like? So it will be look like in this form. One thing more, it is the envelope virus. Fine. So it contains envelope. So now let me draw the structure here. This blue one, as I draw, this one is the envelope side. Fine. With envelope, there are the spikes protein arises. So now let me draw here the spikes protein, or these are the envelope area. Within the envelope, they contain the capsid. Now this green one represents the capsid, and within the capsid, they have the RNA, two strands of the RNA. Fine. Now these two strands are messenger RNA, or we can say two single-stranded, means fine. So they have two single-stranded RNA and they have some necessary enzymes. So now let me draw this enzyme. This black one are the integrase. Integrase. This red one are the protease, and this blue one are the reverse transcriptase. RT means reverse transcriptase. They contain the two messenger RNA, integrase, reverse transcriptase enzyme, and protease enzyme. And with this outer layer envelope, means with this envelope, these envelope have spikes proteins. So now let me draw these spikes proteins. These are the spikes proteins which arises from this envelope area. And these spikes proteins are responsible to attach their cells, to attach the HIV virus on the host cell receptor. Once they attach, so they will be easily internalized or they will be easily engulfed. So what happened after that? Now these spikes proteins are actually made up of two glycoproteins. The black one is called GP120, while this green one is called GP140, and these are actually what? These are the spikes. Fine. These are the spikes. They contain GP140 and GP120 proteins. These are the necessary proteins which are responsible to attach on the host cell receptor. That's why these proteins are very necessary. This is the structure of the HIV virus. Fine. So this is the structure of the HIV virus. Now how they start their life cycle? First, they will present in the environment of the, means they will present in the bloodstream. Once they reach to a suitable area, means once they reach to the host, means to the one thing more, they attack on a T lymphocyte cell, fine, or we can say they attack on T4 cells, fine. So once they reach to a T4 cell, so they will attach their self with the help of these spikes, and then they will be internalized. So they will attack on the, let me draw the host cell here. They have also some receptor. Now let me draw first of all the receptor, how the receptor actually look like. And these receptors are called CCR5. Now these are the receptors of the host cell, means T4 cell. Let me write T4 or T lymphocyte. T lymphocytes. Fine. So what happened? First of all, the host, means first of all the HIV virus. Let me draw the HIV virus here in a small. First of all, the HIV virus receptor. Now this is the receptor of the HIV virus. They will internalize, or this host cell receptor will recognize the receptor of the HIV virus. So they will recognize the receptor of the HIV virus. Once they recognize the receptor of the HIV virus, then what happen? Then this GP140, this one, the green one, GP140, will unfold themselves. These are actually hydrophobic sites. Okay. Now this hydrophobic site means this GP140 will unfold itself. So what happened? Once they will unfold itself and they are hydrophobic in nature, so they will be easily attached. So what we get? We get the virus structure just like that. So now let me draw here. These one receptors will be easily attached because these are the hydrophobic in nature, and once they will attach, so the receptors, so this GP140 receptors will move on the opposite side. Once they will move, means they will move on the opposite side. Once these receptors will move on the opposite side, so the so the virus will easily be internalized. So how the virus will be look like? The virus will be look like in this form. So now the virus will easily be internalized, and once they will internalize, so what they will actually produce? They will produce their mRNA, means 2 strands of the mRNA, and they will release the reverse transcriptase. These blue one as I draw here, these are the reverse transcriptase enzyme. They will also release the integrase enzyme, and as well as they also release the protease enzyme. Fine. Mean this triangle one represent the protease, this square one represent the integrase, and this circle represents the reverse transcriptase enzyme. Fine. Let's suppose these geometric shapes will represent the different enzymes. Now what happened? Once these necessary materials will be internalized inside the host cell, so what happen? Reverse transcription process will happen. Why the reverse transcription process will happen? Because the HIV genome should have integrate with the host genome. Why? Once they will integrate with the host genome, so all the necessary protein and all the necessary material for the HIV viruses are made. So for this purpose, the HIV genome should integrate with the host genome. So that's why the reverse transcription process will occur, and this reverse transcription process will be possible with the help of this reverse transcriptase enzyme. Now in reverse transcription, what happened? The messenger RNA, mean the first strand, is used as a template, and this strand will produce the DNA strand, single-stranded DNA strand. Fine, with the help of this reverse transcriptase enzyme. Now let me explain what happened. First of all, this strain, they will pass their self to the reverse transcriptase, and what happened? And then we get a DNA strand. This red one represent a new DNA strand. but we get the RNA DNA hybrid. Now this RNA DNA hybrid are actually in unstable form. Fine, and they are not unstable. So what happened in this case? The DNA in this case, or this double stranded, or we can say the RNA DNA hybrid will pass this. Actually the reverse transcriptase enzyme have two activity. Fine, so first activity is actually, let me write here, the first activity of this reverse transcriptase is the polymerization, and the second activity is the RNA. Fine, so they have two activity, polymerization and RNase activity. First of all, the polymerization process occur, by which we get the DNA strand. Once we get the DNA strand, so this DNA and RNA hybrid will again passes from this RNA from this reverse transcriptase enzyme, and then RNase activity will happen. Now what happened in RNase activity? In RNase activity, this strand, means the blue one, RNA, messenger RNA strand, will be dissociated. So this site, means the messenger RNA will be dissociated, and this DNA will be around their self to another DNA, and then we get a double stranded DNA. Fine, so here a new DNA will be formed. So these DNA will means the new DNA and the old DNA are complementary to each other, and finally we get the double stranded DNA. So now this double stranded DNA are in stable form, and this double DNA will be easily integrate with the host genome. Fine, so after that what happened? They will goes to the second enzyme, and this second enzyme actually integrase. Once they will passes to the integrase, so they will cleaves the 3' end of each DNA. So if I draw the DNA structure in a linear form, so what we get? Yes, this one. Fine, so first of all, what happened? Try to understand the concept. What happened first of all? The integrase enzyme will comes here, and they will cleave this 3' end. Let's suppose this one, the 3' end. They will cleave this 3' end of the both strand. Fine, so here also a 3' end will cleaved. So once they will cleave the 3' end of each DNA strand, then we get a compatible end of the DNA, and we need this compatible end, or we can say we need these sticky ends. We also call that sticky ends. Fine, so now let me write here sticky ends. Fine, so actually we need these sticky ends. Fine, so once these sticky ends are produced, so these means this DNA will be easily joined with the host genome. So then the integrase enzyme, means here the integrase enzyme will sit here, and they will migrate this DNA to the host genome. First of all, they will, now this one, if I draw the structure of the nucleus here, so let's suppose this one the nucleus. Fine, so these are the nucleus pores. So this DNA will reach to the nucleus with the help of the integrase enzyme. They will carry the DNA, and they will internalize the DNA inside the nucleus. Now inside the host nucleus, there are also the genome of the host are also present. If I draw the structure of the host genome, so let me draw the structure of the host genome with this black one. Now this one, the host genome. Fine, what happened? This genome will also integrate with the host genome. So the host genome will also integrate with the HIV genome. Once the integration process is done, then what happens? Inside the nucleus, there are also the RNA polymerase are present. Means the RNA polymerase enzyme are also present. Then what happens after that? After that, transcription process will occur. So these things will be converted, means this genome of the host will be converted into a messenger RNA. So we get a messenger RNA, means with the help of transcription. With the help of transcription, we get the messenger RNA. Fine. Now this messenger RNA have special codons, and these codons are responsible for amino acid, and then we get a proteins. After that, translation process will occur. And what we get large protein component. Now these proteins are actually in a very large form, and they have a segments just like that. These proteins are very large proteins, and these, we can say, these are not the structure protein, let's suppose, and these are very large proteins. The virus didn't need this protein. So what do the virus? The virus will release the protease enzyme, or this protease enzyme then cleaves these proteins into a small structural proteins. And then the virus will easily form the capsid with these small structured proteins. Fine. So after that, what happened? Protease enzyme will come. So they will pass it with the protease enzyme. Fine. So now here are the protease enzyme. Fine. So now once they will pass it with the protease enzyme, so then we get the structural protein here. These are the structural protein. Then finally we get what? We get the structural proteins. Once we get the structural proteins, then the process will be finished. Not whole process will be finished. Means the capsid formation process will be finished. Then after that, what we get? We get the capsid. From these things, we get the capsid. Fine. Within the capsid, our messenger RNA are also packet their self, means our messenger means the messenger RNA or the HIV. Fine. So the HIV messenger RNA are also packed, and here the three enzymes are also packed. The square one means the integrase, the circle means the reverse transcriptase, and the triangular means the protease enzyme will also packed around inside this capsid. And now this capsid don't have the envelope. Now where they get the envelope? After the capsid portion of the HIV once formed, then they will migrate to the endoplasmic reticulum, and from this endoplasmic reticulum they will get the envelope, and other spikes proteins, mean the spike protein GP140 and GP120 proteins are also they will get from the endoplasmic reticulum. So they will reach to the endoplasmic reticulum. So now let me draw the endoplasmic reticulum. I hope you will see the structure of the endoplasmic reticulum. Usually endoplasmic reticulum are present in just like in this form. Endoplasmic reticulum. In short form we write ER. Now once they will pass from the endoplasmic reticulum, then what we get? Then we get the complete structure, or we get a mature HIV virus. Now this is the mature HIV virus. Fine. There are those two strains. There are the enzyme. And from this endoplasmic reticulum, they also get the spikes protein. Finally, we get the mature HIV. And now this mature HIV will put pressure on the host cell, and then they will release out from the host cell through a budding process. because I already discussed that the envelope viruses are released from the host cell through budding processes, fine, and they will be internalized through a membrane fusion, fine. So they will release out from the host cell. Once they release from the host cell, then they will ready to attack another T lymphocyte or another T4 cells, and the cycle will be started again and again. So that's how they will start their life cycle, means they will start their life cycle, fine. So that's all about the life cycle of the HIV viruses. So now let me explain the whole process in Urdu what happen, और in हिंदी लैंग्वेज what happen. ये आपके पास HIV वायरस का स्ट्रक्चर है. इसके ये जो आउटर साइड आपको दिखाई दे रहा है ये स्पाइक प्रोटीन्स है, सही. ये स्पाइक प्रोटीन असल में ये दो चीज़ों से बने होते हैं, एक GP140 प्रोटीन और एक GP120 प्रोटीन से बने होते हैं, सही. ये आपके पास जो आउटर लेयर है ये आपके पास इन्वेलप है, ठीक है. ये आपके पास जो ग्रीन वाला राउंड एरिया है, ये सर्कल एरिया जो ग्रीन वाला है, ये आपके पास क्या है, ये आपके पास कैप्सिड है. इस कैप्सिड में आपके पास दो स्ट्रैंड होते हैं मैसेंजर आरएनए के. रिवर्स ट्रांसक्रिप्टेज एंजाइम इसके अंदर होता है आपके पास. इसके अलावा इंटीग्रेस एंजाइम होता है और प्रोटीएस एंजाइम होता है. अब ये आकर इस टी लिम्फोसाइट सेल पर अटैक कर लेते हैं और टी लिम्फोसाइट सेल के ऊपर भी CCR5 रिसेप्टर्स होते हैं. और फिर ये जो GP120 होता है और CR5 होते हैं, ये जब यानी एक दूसरे के साथ ये जब मीन्स अटैच हो जाता है एक दफ़ा, तो फिर ये वाले एरिया जो है ख़त्म हो जाते हैं. और ये जो हाइड्रोफोबिक एंड है, मीन्स ये GP140 हाइड्रोफोबिक एंड्स होते हैं, ये फिर अटैच हो जाते हैं इस होस्ट सेल के साथ. जब ये अटैच हो जाते हैं तो फिर ये जो हाइड्रोफोबिक एंड्स होते हैं ये एक दूसरे को अपोजिट साइड पर पुल करते हैं, जिसकी वजह से ये वायरस जो है आसानी के साथ अंदर चला जाता है. और ये सिर्फ यहाँ पर अपने ये जो मैसेंजर आरएनए और ये जो 3 एंजाइम होते हैं ये रिलीज़ कर लेता है, और जो कि इसका ये आउटर एनवेलप एरिया होता है ये बाहर रह जाता है और बाहर ही ख़त्म हो जाता है. अब ये जो मैसेंजर आरएनए होता है ये इस एंजाइम से चला जाता है, मतलब रिवर्स ट्रांसक्रिप्टेज एंजाइम में जाता है. और फिर हम क्या हासिल कर लेते हैं, हम एक DNA स्ट्रैंड हासिल कर लेते हैं. अब इसके बाद हम आखिर में DNA RNA हाइब्रिड हासिल कर लेते हैं. ये अनस्टेबल फॉर्म में होते हैं. ये DNA RNA हाइब्रिड दोबारा इस RNA रिवर्स ट्रांसक्रिप्टेज में चला जाता है और फिर क्या होता है, हम आखिरकार एक नया स्ट्रैंड, ये जो ग्रीन कलर का है, नया स्ट्रैंड हम DNA हासिल करते हैं. तो हम एक कंप्लीट डबल स्ट्रैंडेड DNA हासिल कर लेते हैं. असल में हमें इसी डबल स्ट्रैंड DNA की ज़रूरत होती है क्योंकि ये आसानी के साथ इस होस्ट जीनोम के साथ मतलब आसानी के साथ इंटीग्रेट हो सकता है. तो क्या होगा फिर, ये यहाँ पे एक दूसरा एंजाइम है जो कि इंटीग्रेस है. इंटीग्रेस क्या करेगा, इस DNA के जो 3 प्राइम एंड होते हैं उसको कट कर लेता है. जब इस 3 प्राइम एंड को कट कर लेते हैं तो हम DNA को बिल्कुल इसी तरह हमारे पास DNA मतलब आ जाते हैं. ये मैंने लीनियर स्ट्रक्चर ड्रा किया है, तो DNA तकरीबन इस तरह, मीन्स हमारे पास स्टिकी एंड आ जाते हैं. इस स्टिकी एंड की ज़रूरत असल में हमें पड़ती है. क्यों? क्योंकि जब ये स्टिकी एंड होगा तो ये DNA आसानी के साथ होस्ट के जीनोम के साथ मिल जाएगा. स्टिकी एंड के प्रोडक्शन के बाद इंटीग्रेस इसके ऊपर बैंड हो जाते हैं और फिर ये इसको कैरी करके न्यूक्लियस के पोर में चला जाता है ये DNA, और फिर ये होस्ट के जीनोम के साथ बाइंड हो जाता है. तो ये देखो, ये ब्लैक वन होस्ट जीनोम है और ये जो मिड वाला एरिया है ये HIV का जीनोम है. जब ये प्रोसेस हो जाता है इंटीग्रेशन का तो फिर हमारे पास इससे ट्रांसक्रिप्शन का प्रोसेस होता है. मींस हमारे पास यहाँ से मैसेंजर आरएनए बन जाता है, क्योंकि यहाँ पर ख़ुद भी न्यूक्लियस के अंदर आरएनए पॉलीमरेज एंजाइम होता है. तो वो आरएनए पॉलीमरेज ट्रांसक्रिप्शन का प्रोसेस करता है. फिर इसके बाद जब ट्रांसक्रिप्शन का प्रोसेस हो जाता है हमारे पास मैसेंजर आरएनए प्रोड्यूस हो जाता है. इस मैसेंजर आरएनए से फिर क्या होता है, इस मैसेंजर आरएनए से फिर हमारे पास प्रोटीन्स बन जाते हैं. ये प्रोटीन्स आपके पास कॉम्प्लेक्स लार्ज होते हैं. तो प्रोटीएस एंजाइम आकर इस प्रोटीन को स्मॉल सेक्शन में कन्वर्ट कर लेते हैं. तो वहाँ पर हमारे पास स्ट्रक्चरल प्रोटीन्स बन जाते हैं. ये वाले ये स्ट्रक्चर प्रोटीन कैप्सिड एरिया भी बना लेते हैं और इस कैप्सिड एरिया के अंदर फिर ये तमाम तर ये जो सिंगल स्ट्रैंडेड ये स्ट्रैंड और यहाँ पे जो हमारे पास मीन्स 2 मैसेंजर आरएनए जो होते हैं वो भी अंदर चले जाते हैं और ये जो 3 नेसेसरी प्रोटीन है तो वो भी अंदर चले जाते हैं. लेकिन अभी तक हमारे पास ये जो सेल है इसके ऊपर सेल नहीं, ये जो वायरस है पार्टिकल है इसके ऊपर अभी तक इन्वेलप नहीं है. तो फिर क्या होता है, ये इन्वेलप यहाँ इस एंडोप्लाज्मिक रेटिकुलम से हासिल करते हैं. तो ये फिर एंडोप्लाज्मिक रेटिकुलम किससे पास होता है, यहाँ से जब ये निकलता है तो इस पर इन्वेलप भी होता है और स्पाइक्स भी होते हैं, मींस ये ग्लाइकोप्रोटीन्स यानी GP140 और GP120 भी होते हैं, तो आखिरकार हमारे पास एक मैच्योर HIV बन जाता है और फिर ये होस्ट सेल से निकलते हैं बडिंग के ज़रिए। सही। So that's all about the life cycle of the HIV viruses. I hope this video helpful. If you understand, so like the video. If you have questions, so write it into a comment. I will try my best to give you answer. So please like the video, share the video, and subscribe the channel to get more interesting video lecture like that. Thank you so much.